Hello everyone. I'm welcome again to this new course of plant physiology and metabolism. In this lecture today we are going to discuss about the uh, role of phosphorus in detail, its uptake, role, and then deficiency symptom and toxicity. So you see that phosphorus has very varied aspects. Its essentiality in plants is very important. Then how phosphorus is taken up by the plants. Then we will discuss in detail about the important role played by the phosphorus in plants. Then specific deficiency symptoms and then toxicity symptoms. So you see that phosphorus plays very important role and it's a macronutrient essential for plant growth and development, just like nitrogen we have discussed in our previous lecture. So plants basically absorb this nitrogen, plant absorb phosphorus from the soil in different forms depending upon the pH of the soil, and most importantly phosphorus is taken up by the plants in the form of orthophosphate and phosphate, uh, which occurs in the soil solution at very low concentration, that is 0.1 to 10 micromole. So it's very important to uh, share this fact that most of the cations except sodium, potassium, ammonium and lithium react with phosphorus to form insoluble salts, rendering phosphorus unavailable for the plant growth. So what is the way out? So in soil that is rich in iron and aluminum, most phosphates they remain inaccessible to the plants. However, with the addition of chelating agents generally helps to release inorganic phosphate from aluminum and iron. The importance of phosphorus can be just further strengthened with the help of this figure. You see that in the slide, you see there are two plants. One is having long root, that is A. So another plant having well developed root. So basically this phosphorus deficiency affects the root growth in plant, especially the primary roots. Elongation of primary roots is generally inhibited. So however, the density of lateral roots and root hairs generally increases. So this is sort of compensatory mechanism by the plants to improve phosphorus uptake from the soil. Then you see that in another figure you see that how plants adapt to increase phosphorus availability when it is deficient. So they generally do it by secreting substance like phosphatase, organic acid and protons into the soil, which help to break down organic phosphorus compounds and make phosphorus more accessible to the roots of the plant. In addition to this, plants can also increase phosphorus uptake through specialized transporters called phosphate transporters. Another method which could involve forming associations with beneficial fungi called vesicular arbuscular mycorrhiza, abbreviated as VAM, which develop a symbiotic relationship with the plant's roots and extend generally a thread-like hyphae into the soil and enhance the uptake of phosphorus, especially in acidic soils that are low in phosphorus. So these basically mechanism help plant to cope with phosphorus deficiency and make sure that they can access the nutrients they need for the growth and development. Now come to the functional role of the plant. What is the important role played by the phosphorus? Uh, you see that uh, phosphorus plays a very crucial role in the macromolecular structure, particularly in the nucleic acids, DNA, and RNA, which are responsible for translation of genetic information. You see that in both DNA and RNA, phosphate acts as a bridge between ribonucleoside units, facilitating the formation of macromolecules. You see in the figure. In addition to this, phosphate also contributes to the highly acidic nature of the nucleic acids, resulting in the elevated cation concentration generally found in RNA and DNA. And the ratio of phosphorus in RNA to the total organically bound phosphorus varies across tissues and cells, but it is very important to share that notably high in expanding leaves, generally the concentration of phosphorus is very high in expanding leaves where there is substantial demand for ribosomal RNA to facilitate rapid protein synthesis. In contrast to this, this ratio is lower in mature leaves and significantly reduced in senescing leaf. So, the bridging form of phosphorus diester, the bridging form of phosphorus diester is also very much prevalent in phospholipids that is found in the biomembranes. And then what happens, what is the role within biomembranes? Basically in biomembranes, this phosphorus acts as a bridge between a diglyceride molecule and with another molecule which could be an amino acid or amine or alcohol. In biomolecules, basically amine choline commonly serves as a primary partner, resulting in the formation of phosphatidylcholine, also known as lecithin. But you see that the majority of the phosphate esters serve as intermediates in metabolic pathway involved in both biosynthesis and degradation. And their formation and function are basically closely linked to cellular energy metabolism and energy rich phosphates. For instance, the energy needed for the process like starch biosynthesis or in ion uptake, that is provided by energy rich intermediates or coenzymes, and most importantly ATP. Now, what is the role of phosphorus in energy transfer reaction? As you see that phosphorus is a very vital component of ATP. You see in this diagram. So, this is the structure of ATP molecule, and the ATP is also known as energy currency of the cell, and you see that it is a special carrier of free energy that occurs in protoplasm in free state. And chemically ATP is basically a nucleotide consisting of adenosine, that is adenine plus a ribose, and 3 phosphoryl groups linked by phosphodiester bond followed by 2 phosphoanhydride bond. So, starting from ribose, you see that the phosphoryl groups are designated as alpha. This starting from this ribose, this phosphate is known as alpha, this one is beta, and this one is gamma. And basically these two bonds are high energy bonds, or you can say energy rich bonds. So, once these two bonds are broken down, a high amount of energy is released from the conversion of ATP into ADP. You see in this here how much energy is released, 7.3 kilocalorie per mole energy is released by breaking this phosphoanhydride bond. These two bonds are same, and this one is ester bond. So, basically this ATP molecule has one ester bond and two phosphoanhydride bond. Now coming, what is the role of phosphorus in generation of ATP? You see that ATP is generally consumed and then it is further produced. So the regeneration of ATP from ADP and phosphate is called phosphorylation, and this phosphorylation is basically an endergonic process and which could take place in three different ways. One important one is substrate level phosphorylation. So question is, what is substrate level phosphorylation? So you see in this, ATP is formed by direct transfer of phosphoryl group from a high energy rich compound to ADP without the involvement of electron transport system. A very interesting example in this regard of substrate level phosphorylation is uh, conversion of phosphoenol pyruvate into pyruvate and accompanied by uh, formation of ATP from ADP. So you see that in fact uh, some compounds in biological system have high energy phosphoryl transfer potential than that of ATP. So means these compounds have high phosphoryl potential than you see from ADP, and these compounds are phosphoenol pyruvate, 1,3-bisphosphoglycerate and creatine phosphate. These three molecules have more phosphoryl group transport potential than ATP, and you see that the other molecules like glucose 1 phosphate, pyrophosphate, glucose 6 phosphate, glycerol 3 phosphates, these compounds have less phosphoryl group transfer potential than ATP. So basically what is the important aspect I would like to share, that you see in this given example, conversion of phosphoenol into pyruvate. So what basically phosphoenol pyruvate does? Phosphoenol pyruvate can transfer its phosphoryl group to ADP to form ATP, and it's very significant to note here that ATP has a phosphoryl group transfer potential that is intermediate among the biologically important phosphorylated molecules. So basically this intermediate position makes ATP to function efficiently as a carrier of phosphoryl groups. Now another method of ATP generation I will discuss in detail while discussing the respiration. Just a basic outline. Generally oxidative phosphorylations occur during aerobic respiration in mitochondria, and in this generally ATP is formed and which is linked to the terminal oxidation of coenzymes like FADS2, and FADS2 is further oxidized through respiratory chain that produce 2 ATP, and the oxidation of NADH produces generally roughly 3 ATP. Then photophosphorylation I have discussed in my previous lecture. Basically ATP is synthesized during the light driven electron transport in the thylakoid membrane of chloroplast. So in higher plants it occurs by cyclic and non-cyclic pseudocyclic processes, but in purple and green bacteria, due to absence of photosystem 2, only cyclic photophosphorylation occurs. So now what is the role of phosphorus in modulation of enzyme activities? You see in this figure that enzyme is basically in inactive state, and but phosphorylation of enzyme proteins by ATP, GTP or ADP is sort of mechanism through which energy rich phosphates regulates enzyme activity. By attaching with the enzyme, it regulates the activity of the enzyme. So this regulatory phosphorylation is generally facilitated by protein kinases and which lead to the activation, inactivation and or alteration in the allosteric properties of the target proteins. So basically dephosphorylation on another side typically involves hydrolytic reactions catalyzed by another enzyme, phosphatases. So protein phosphorylation is a very crucial element in signal transduction pathways such as those mediated by the phytochrome in plant different responses. Phosphoenol carboxylase, you remember that it's a very important enzyme in C3 and C4 plant, is generally regulated by phosphorylation. In addition to this, in C4 and C4 and chyme plants, phosphorylation generally increases the activity of phosphoenol pyruvate carboxylase while simultaneously reduce its sensitivity to negative feedback controlled by high malate concentrations. Therefore, pyrophosphate is very essential in regulating various activities of enzymes. Now, what is the role of phosphorus as a regulatory role? It's very important that many enzymes reactions generally you see that a phosphate is either serves as a substrate or an end product, like once ATP is converted into ADP and phosphate. And in addition to this, phosphorus also controls some of the key enzymatic reactions. And compartmentalization of therefore phosphate is very essential for the regulation of metabolic pathways in the cytosol and chloroplast. You just see as an example, phosphorus is released from vacuoles into cytosol and stimulate the activity of phosphofructokinase. This enzyme is involved in glycolysis, and as you see that this enzyme regulates a very key reaction and regulates the flux of substrate in the process of glycolysis. Now we see that how this phosphate regulates the activity of certain enzymes, that basically this inorganic phosphate acts either as a substrate or an end product, for example in the conversion of ATP to ADP and phosphate. And in addition to this, phosphorus plays a regulatory role in some key enzymatic reaction, and you see that therefore phosphorus is very much crucial for the regulation of metabolic pathway in both cytosol and chloroplast. For example, in fruit tissues of tomato, phosphorus is released from vacuoles into cytosol and which can stimulate the activity of phosphofructokinase, that is a very key enzyme involved in regulating substrate flux in glycolysis. So basically this release of inorganic phosphate from vacuoles can initiate the respiratory burst during the fruit ripening, highlighting the importance of inorganic phosphate compartmentalization in orchestrating metabolic processes during plant development and ripening. So you see in this slide that glucose is converted into glucose 6-phosphate, but you see that the activity of enzyme phosphofructokinase is inhibited by ATP and citrate. So you see here the enzyme activity of glucose hexokinase is inhibited by glucose 6-phosphate, whereas the activity of phosphofructokinase is inhibited by ATP and citrate. But another reaction you see that another enzyme phosphofructokinase 1 is generally activated by adenosine monophosphate and fructose 2,6 by bisphosphate. So these two molecules they serves as an activator of phosphofructokinase 1, whereas in the another reaction you see that you see the enzyme pyruvate kinase which is further inhibited by ATP. So, and in addition to this, activity of pyruvate kinase is activated by fructose 1,6 bisphosphate. So, you see that how compounds containing phosphorus serve as a switches to regulate a very important metabolic process, that is glycolysis. And you just recall that in the process of photosynthesis and the allocation of carbon during the light dark cycle are significantly influenced by the concentration of inorganic phosphate within the stroma of the chloroplast and partitioning between chloroplast and the cytosol. So, you just might remember in the presence of light, optimal photosynthesis requires a inorganic phosphate concentration in the chloroplast ranging from 2 to 2.5 millimole. Opposite to it, photosynthesis is severely inhibited when the inorganic phosphate concentration drops below 1.4 to 1 millimole. So, the demand for inorganic phosphate is particularly high for phosphorylated intermediates involved in photosynthesis, and consequently in leaves of the plants deficient in phosphorus, where there is a lack of vacuolar buffering, inorganic phosphate concentration may decrease by up to 50% following the onset of light. So, basically this reduction in availability of phosphorus can significantly impact the efficiency of photosynthesis and overall productivity. Now, role of phosphorus in phytate and polyphosphate formation. So, when the supply of phosphorus increases from deficient to sufficient levels, the concentration of major phosphorus fraction in vegetative plant organs also increase. So, with further increase in phosphorus supply, only inorganic phosphate serves as a major storage form of phosphorus in highly vacuolated tissue, which further increases. Therefore, plant can store phosphorus in two other major forms. One is phytate and inorganic polyphosphates. So, the storage of phosphate in cells as inorganic polyphosphate is very common among bacteria, fungi, green algae and higher plants. So, basically plant synthesized polyphosphates are linear polymers of inorganic phosphate with pyrophosphate linkages and that are energetically equivalent to ATP. Polyphosphates may also serve as energy storage compounds and regulate the inorganic phosphate concentration in the metabolic pool of the cells. They also function basically as a cation exchanger, such as potassium in the chlorella and calcium in mycorrhizal fungi. And in addition to this, phytate serves as a typical storage form of phosphorus in grains and other different seeds. And basically phytates are salts of phytic acid, myoinositol, and it is synthesized from the cyclic alcohol myoinositol through esterification of hydroxyl group with phosphoryl groups. So, this is the role of uh, phytate in plants. Now, come to the deficiency symptom of induced by phosphorus. You see that deficiency of phosphorus also leads to an increase in the activity of enzyme phosphatase. So, increased activity of enzyme phosphatase generally leads to release of phosphorus from organic compounds. However, generally what happens, this is coupled with inhibition in cambial activity, which is responsible for the growth of vascular tissues. Additionally, you see that it has been noticed that there is a decrease in protein synthesis in response to deficiency of phosphorus. Those plants that lack enough phosphorus, and it has been noticed that their leaves do not grow as big as they should be, and they might not have as many as overall leaf in the plants. So interestingly, what have been noticed that when plants don't get enough phosphorus, their roots tend to grow more compared to their shoots, and this means that the ratio of roots to shoot varies. And one important visible effect of phosphorus deficiency is the appearance of dead patches on the leaves and fruits, and the leaves themselves may not change the color, showing the signs of discoloration as you see in this slide. And very interesting observation, you might have observed that you see that purple discoloration appears in the leaf. So why this leaf appears purple in plants facing deficiency of phosphorus? So basically uh, during the deficiency of phosphorus, plant respond by producing anthocyanin, which is a pigment that gives leaves a reddish color, but this is often followed by the aging process known as senescence. So in some plant species, it has been noticed that there is a formation of specialized root structures called clusters or daciform root clusters, which generally help the plant to acquire more phosphorus from the soil. So overall, the phosphorus deficiency could also affect the reproductive capacity of the plants, and generally flowering is delayed, and you see that formation of seed is also hampered, and additionally the leaves of these plants tend to age prematurely, which further limits their ability to produce seeds. So you see that phosphorus imparts very important role, and its sufficiency is important for normal growth and development of plants. So you must be clear that just mere one example I have shown in maize leaves how deficiency symptom of phosphorus appears, and there are other plants you try to identify that how phosphorus deficiency could be identified in other plants on the basis of knowledge you gained regarding the functional role of phosphorus in plants. Thank you very much.